when you see a question, if it says phenotype, you have to give me the phenotype, which is yellow, green. If I ask you genotype, you have to give me big Y, or big Y, little y, like that, right? And if it's a heterozygote and you see a phenotype, that is the dominant phenotype. So in this case, the heterozygote is big Y, little y, and you see a yellow color, which means that yellow is the dominant color, right? That's the dominant allele, the big Y. Yeah. Okay. Sometimes we are used to writing it like this, which means, and we say yellow, which means that there is any option. There are multiple options for this dash. It could either be big Y, or it could be big Y, little y. Either one will give you the yellow, right? Can you all see that? Okay. Everybody got it? So let's move on. So a lot of the slides are redundant, the same information, but I want to make sure that I spend, at least for the first lecture, enough time going over these concepts so that it's all clear to you, okay? Okay, so then this one is showing you again the same thing, genotype versus phenotype and homozygotes and heterozygotes. So I'm actually going to go over this very quickly from this cross, which is a dihybrid, big Y, little y. I mean monohybrid, but both parents are heterozygous, big Y, little y cross to big Y, little y. You have the 1:2:1 genotypic ratio and 3:1 phenotypic ratio. Genotypic ratio is going over this again. Genotypic ratio is looking at the genotypes. So it's big Y, little y cross to big Y, little y. The genotypes are big Y, big Y, big Y, little y, big Y, little y, and little y, little y. You may do a Punnett square if you want to, but you should not have to for small little things. You should be able to tell just by looking at it. So this is 2, this is 1 and 1. So this is your 1:2:1 ratio, right? And phenotypes are all these three are yellow, and this is 1. So this is 3:1, yellow to green, right? Okay. Next slide. There's a poll question. I'm going to quickly do this poll and see how you guys are able to answer this. So I'm going to share this poll and I want you guys to answer. Well, I'm going to check the chat if there are anything that needs my answer. Okay, I'm going to stop in just a minute, not a minute, a few seconds. Okay, I'm going to end this poll, share the results. Most of you got it right, but some of you still got it wrong. I want to make sure everybody can answer these types of simple questions. So you have to read the question carefully. You just write it down here. So you have round peas are dominant and wrinkled peas are recessive. You cross to homozygous dominant, so it has to be RR. Cross to a homozygous recessive, so it has to be RR. So what would be the offspring be? RR. And what would they be? Round, because round is dominant. So it's round and RRB is the correct answer. Okay. Okay, let's go to the next slide. So dominant relations prediction of phenotype from genotype. That of course we can do if we know which one's dominant and which one's recessive. Homozygous versus heterozygous, phenotypic ratios versus genotypic ratios, and applying the laws of probability. So we've done all of these. Let's just talk about this step by step. So we have test crosses. So that's a term that we have seen before. A test cross is basically any sample or any individual crossed to a homozygous recessive. So in this case, you have a yellow, let's say a yellow plant, crossed to a recessive homozygous plant. Okay, this is called a test cross. Test cross is always a plant whose genotype you don't know crossed to a homozygous recessive, right? So if you did this cross, and I'm going to show you the outcomes here. Just you can use this while you're also following my scribbling here. So when you cross this yellow with a homozygous recessive green, there are two options for this yellow. One option is a big Y, and the other option is a big Y, little y. When you cross this with this, what you will get is, if you will be able to see it, yeah, what you will get is Y only. This is the outcome, and it will be yellow. All offspring will be like this. Instead, when you use this cross, so let's say your yellow is this, you will get all yellow. But if your yellow is this, what will be the outcome? You will get this, plus you will get this in a 1:1 ratio, right? So based on that, looking at the phenotype, if you did a test cross, you can figure out if you get all yellow, then your yellow was this, original yellow. If you got 1:1 yellow to green, then your original yellow was this, right? A test cross can reveal an unknown genotype. Everybody follow this? You guys, I see that a lot of other stuff is going on the chat. When you're, if you, do that for like a few minutes is okay, but not throughout the lecture. I need you guys to focus on what I'm saying, okay? So otherwise I would have to disable chat with everybody. So don't do that anymore. Just listen to what I'm saying, understand it, because that's more important, okay?
Professor, I'm sorry. I have a question.
Yeah.
So the test cross is, it applies to be able to determine if the unknown is homozygous or heterozygous?
Unknown genotype, yes.
Okay, thank you.
Okay, all right. So now we move on to dihybrid crosses. We talked about monohybrids, which was looking at one genotype or one trait at a time. For dihybrids, we're looking at multiple traits. So dihybrid, we're looking at two traits, and then you have multihybrids where we can look at multiple traits simultaneously. So let's try a dihybrid looking at two traits. A dihybrid means that you have two different phenotypes, and hybrid means they're heterozygous. So you have YY and RR, so yellow and round. Okay. So if you crossed, as is shown here, if you cross, let me try to get the pointer. If you crossed a homozygous pure breeding yellow round with a homozygous pure breeding green wrinkled, you will get all F1s, which will be heterozygous for the yellow and heterozygous for the round. Everybody with me? When you self cross this or cross this to itself, you have a big Y little y, big R little r cross to a big Y little y, big R little r. So this is a dihybrid cross, and what you end up with is your ratio, which you know, all of you know this ratio, right? You get a ratio, phenotypic ratio of 9:3:3:1, and then this is yellow and round. My screen is not large enough, so, and this one is yellow and wrinkled. This one is green, round, and this one is green, wrinkled. Okay? Everybody see this? So that's how Mendel did his experiments, and he always repeatedly came up with this ratio. So Mendel asked whether all the F2 progeny would be the parental types, yellow, round, and green wrinkled, or would they be recombinant types? So these are called the recombinant types because these were the ones you started out with. This was yellow wrinkled and yellow round, and this one was green wrinkled. So those were the parental types, and these are the recombinant. And this one's the parental types. Okay, so that's what Mendel tried to do, and what he found was you get all of these options, and therefore what he concluded was his second law, which was the law of independent assortment, which we will talk about in a little bit. So here, let's see how this works. So you have, starting with the parental, remember when we put that dash, it means it could be anything. But you know, the phenotype is yellow and round. Here the phenotype is green and round. Here the phenotype is yellow and wrinkled. Phenotype is green and wrinkled, right? So ratio, if you just look at one trait alone, ignoring the other trait, okay? You can always do that with a dihybrid, is just look at one trait ignoring the other trait. Then you should get the same 3:1 ratios. So if you just look at how many are yellow, you add up the 315 and the 101. And those are the yellows, and how many are greens? 108 and 32. So that would be 3 times as many yellows as there are greens. If you just looked at the rounds and the wrinkles, the rounds are these and these, and you look at these and these are 3 times as much as these. So you always get the 3:1 ratio of each trait independently of the other trait. Okay, so that's what Mendel's law of independent assortment says, is that each trait behaves independently of the other trait, but in combination, you get both the parental and the recombinant one. So you have every possible combination of both of them. Yeah, I'm trying to keep the whiteboard in the same way that I'm writing, but it's hard sometimes. So yeah, remind me if it doesn't happen. Everybody okay with this material so far? So now you're doing this Punnett square, right? This is what you're used to doing, Punnett square. Independent assortment of the process. So you have each F1 dihybrid produces 4 possible gametes in a 1:1:1:1 ratio. So that's why you can do the Punnett square, right? So this is how a lot of students get confused when they write the Punnett square also, because you want to get one allele of each trait together, because the gametes are formed that way, right? The gametes have only one copy of every chromosome, so therefore only one allele of every trait. So in this case, if you have a YYRR, you will get gametes like this. You will get every possible combination of this YR, 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 and Y. you will get all of those 4 gametes in the ratio of 1:1:1:1. Right? You will get all of these 4. So 2 of them are like the parents and 2 of them are the recombinants in the 4 phenotypic classes that you get, which we saw in the 9:3:3:1, right? So law of independent assortment, genes for different traits assort independently of one another in the production of gametes. And what's important is you can look at each trait independently, ignoring the other trait, and you should still get the 3:1 ratio for the heterozygous cross. And you should be able to combine those two to get your dihybrid ratios, which I'll show you in a minute. So Mendel's law of independent assortment, we went through this already. I just wrote it down here. So you have big Y and big R, or big Y and little r, big little y and big R, or little y and little r, all 4 possible combinations. So now I will show you how, if for this is only 2 traits. If you had multiple traits, you couldn't possibly do a Punnett square. So I'm going to show you how you can use the law of the product or the product rule to combine the 2 traits and get your offspring ratios for both traits combined. Okay. So for the dominant trait, you have, so let's look at this example that they have shown only. So you start with a big Y, big R, little r, and you cross that. Right? You're doing this cross. And if you just separate these out, you just can do this separately. You don't have to keep them together. You can separate them out. And so now this one is going to give you a YY. One is to 2:1. And this one is going to give you a RR. Right? Everybody with me so far? Now, if you want to find out how many offspring are going to be, I'm going to have to move up, how many offspring are going to be, I'm just arbitrarily writing this. How many are going to be like this? 2 out of 4. And then how many are going to be like this? 1 out of 4. So how many are going to be like that? 2 out of 16. Okay. Everybody with me? But if you want to just a phenotype, how many are going to be yellow and round? How many will be yellow and round, right? So yellow are going to be 3 out of 4. Round are going to be 3 out of 4. So that's going to be 9 out of 16. Right? So you don't have to do the Punnett square, you just apply this product rule to get your outcomes. Everybody with me? Any questions?
Professor,
Yeah.
could you show me again how you got 3 out of 4?
For the yellow?
Yes, yes.
So for the yellow, here is the yellows. These are the yellows, right? 2 plus 1, 3. 3 out of 4 are yellows.
Oh, okay. I see. Okay.
Right?
Yeah.
Okay. Everybody good? Okay. Okay, so that was one way to do it, is apply the product rule. The other way is like a branch, 3 branch type things, branched line diagrams. So that's out of this cross, again, I'm going to have to write this up. Out of this cross, you can just do the one yellow first, And you know that 3 out of 4 will be yellow, right? Out of those 3 out of 4, how many will be red? I mean, round. Again, 3 out of 4 will be round, and 1 out of 4 will be wrinkled, right? So how many will be? So this is yellow, this is round, and this is wrinkled. So how many will be yellow and round? Is 3 out of 4 times 3 out of 4. How many will be yellow and wrinkled? 3 out of 4 times 1 out of 4. And then 1 out of 4 is 1 out of 4 is green. Out of that, 3 out of 4 will be round, and 1 out of 4 will be wrinkled. So this is green and round are 3 out of 16, and green and wrinkled are 1 out of 16. Got it? So it doesn't show up there. Can you see that? Right? Everybody with me? Questions? Okay. From the quiz, you can ask me later because a lot of students haven't taken the quiz. Okay? Let's wait. So you can email me separately and I'll be able to answer you. Okay? Okay, so now let's do test crosses for the dihybrids. So just like we saw for the monohybrids, a test cross is crossing an unknown genotype to a homozygous recessive. Okay, so if you had a yellow and a round, which would be an unknown genotype, what are the options for yellow and round? You have options like this. Yellow and round could be like this. Yellow and round could be yellow and round could be like this. And the last one would be, these are the 4 ways you can still have yellow and round, right? Everybody is able to see this? So now, to figure out which of these genotypes is present in your yellow round sample, you do a test cross. Each one you cross with green and wrinkled. Which one of these is your yellow round? So I'm going to not have space to write this down here, so I'm going to just follow what is shown to you here. And so let's look at the first one. Big Y, little, big R, big R. If this was your yellow round, what would happen? You would get all yellow round offspring because see, you have the only gamete possible from here is big Y and big R. And then of course, the only gamete possible from your homozygous recessive is little y, little r. So you will get a heterozygous, all F1s will be this. So if you get only one type of F1s, then that means that you have big Y, big Y, big R, big R. If you have the second option, which is shown here, I'm going to go here, big Y, big R, big R. If you had that option and you did that test cross, the gametes that can be formed from here are big Y, big R, or big Y, little y, big R. So big Y, big R, or little y, big R. So you can get a 1:1 ratio of yellow round or green round, right? So you get two possible outcomes. So this would be your sample that you started out with. If instead you had a big Y, big R, little r, so this one, then when you did your test cross, you would end up with a yellow round and a yellow wrinkle. So that's how you can tell that your original sample was this, right? Yeah, the test cross is always homozygous recessive, okay? So that's what you'll get. If you had a heterozygote, double heterozygote, then you have a big Y, little r, you get 4 possible gametes and you get all 4 possible outcomes. Yellow round, yellow wrinkle, green round, green wrinkle. So then you know your sample is heterozygous. Everybody understood? So how test cost can be applied to dihybrids? Any questions? So then let's do the poll question. Okay. Let's launch this poll. I'm going to give you a couple of minutes. Read this question carefully and then answer. So I'm reading it out for you. In cats, black color B is dominant to a special temperature sensitive albino gene B, little b, which produces cats with dark legs, faces, and tails. Okay, these are called Siamese cats. Short hair big H is dominant to long hair little h. A short haired Siamese colored cat. So a short haired would be what? Think about it. Siamese colored would be what? Genotype. It's crossed to a long-haired black cat. And now they have these kittens, 2 black short-haired, 2 black long-haired, 2 Siamese short-haired. So what are the genotypes of the 2 parents? I'll take this for a minute. I'm going to work on this while you guys work on it as well. Okay, how much time do you guys need? You still need time? I'm going to give you a little more time. Students are still working on it. Think about the test crosses. You know, you can separate the crosses, right? You can separate the color and the hair. We'll stop at 4 minutes and then we will look at the question. Okay, then I'm calling. So I'm sharing the results. The correct answer is B. No, wait. Yeah, the correct answer is B. B, little b, big H, little h, and big B, little b, little h, little h. The correct answer is B. Okay, the reason being that you get a 1:1:1:1 ratio of all 4 genotypes, right? I mean, all 4 phenotypes, all 4 parental as well as the recombinant ones. And therefore, so if I try to work this, if I show you the Oxfam camera, then what you see is this is your cross. And your phenotypes, I'm only giving you the phenotypes here. So those are your outcomes, right? So you basically get, okay, let me give you another trick. So if you just look at this, then you had, so H stands for short hair, right? So short hair was like that, because short hair is dominant. Short haired, Siamese colored cat. Siamese colored is recessive, so that's like this. And bred with a long-haired black cat. Long-haired is recessive, so it's like this, and black is dominant, so it's like this. Everybody with me? Yes? Okay, now you just have to find out what is here. Now you got all 4 types of offspring. You got a black and short, you got a Siamese and short, you got black and long and Siamese and long. You got all 4 options. 1:1:1:1 ratio, right? Which means that you have to have for it to end up. So let's just look at the big H. Can stop to write. Let's just look at the big H cross to the H. It's easy to separate them out, right? So then the only way you can get H, H, and HH genotypes from here is if this was H, right? And the same thing for the other one, you had a BB cross to a B. And the only way you could have got a BB and a BB is if this was a little, right? So therefore you can put down the little h here and you can put down the little b. Okay? Because these, when you separate them out, they become test crosses, right? So then you can tell. So the correct answer was B, okay? Questions, anybody? I'm sorry, I can't hear you.
For test crosses, there needs to be at least one recessive trait.
Yeah, the test cross is any phenotype that you don't know what the genotype is crossed to a homozygous recessive. Okay. Did I answer your question, Deborah?
Yes.
Okay, good. Any other questions? Should I lower the board a bit? Is that what you're asking me, Sarah?
Like, we can't really see the bottom part.
Oh, you want to see the bottom part? This part?
Yes, thank you.
Okay. Anybody else? Let's move forward then. So, okay, we talked about dihybrids. We talked about two different traits at the same time. What about if you had multiple traits that you're looking at? 5, 6 different traits. For every trait, you have the different types of gametes possible are 2. Every trait, there are 2 possible gametes. So if you have 2 traits, then it's 2 to the power 2. That's 4 different gametes possible. If you have 4 traits, then 2 to the power 4, 16 different gametes possible. So when you have 2 gametes, 2 traits, 4 gametes possible, you can do a 4 by 4 Punnett square. That would be 16 different squares to fill in. But if you had 16 gametes possible because there are 4 different traits, there will be 256 squares to fill out, which is hard to do on a Punnett square. So that's why you have to apply those product rule and some rule to actually calculate the outcomes. So let's see how that works. Okay. So I'm going to erase this and start over. Okay, so now if you had a cross like this, remember, because of independent assortment, you can separate each individual trait. I'm not going to write unless I have to. So here you have a cross shown here, big A, little a, big B, little b, big C, little c, big D, little d cross to the same. Okay, what are the proportion of the progeny with big A, little b, big C, and big D, little d? You can separate each one individually. So you can separate this one. So big A cross to big A, little a, you will get, what do you need? Big A. So, okay, I'm going to write. So big A, little a cross to big A, little a, and then you will get, you need the big A. So I'm going to write down the genotypes. AA is to AA. Is to AA is 1, is to 2, is to 1. Are you all with me? Then the second one is big B. For same thing, it will be the same for all. B, C, D, because they're all heterozygous. You need big A. So anyway, I'm going to write it down just so it's easy for you to track. And then CC cross to CC is and then BD cross to R. Right, you can see there are 4 at the same time right now. What we need now, okay, I'm going to have to go up. What we need is big A, which is this one, 1 out of 4. We need little b, which is this one, which is 1 out of 4. And then we need big C, little c, which is this one, 2 out of 4. And we need this one, which is again 2 out of 4, right? You can change this to 1 out of 2 because it will be easier for you to calculate. So you apply the product rule because you want all of those 4 things to happen, right? Are you with me? And so that is going to be 4 times 4 times 2 times 2 is 64. So it's 1 out of 64. Okay. So instead of doing a Punnett square to figure out what proportion of offspring are going to be like this, you can just apply the product rule. Okay. Questions, anybody?
I do, but going back to the test cross on the hybrids, I got confused with the poll question.
Oh,
Did-
the poll question. You want to go back to the poll question?
Yeah, please.
Okay, I erased everything for the poll question. What was your question?
So I just got confused in general. So we write B and then you did the underscore because we don't know what if it will be.
Yeah. So I did it like this because so what I did was I'm going to try and keep that, not erase that. So if so it says a short haired Siamese colored cat. So short haired is dominant. So it will be like this. And Siamese colored is recessive, so it will be like this. So the first parent is like this: a short haired Siamese colored cat is bred with a long head, which is recessive. So we're going to put it like this, and a black cat, which is like this. Are you with me now?
Oh, okay. Yeah. So we're always gonna do recessive to the what's known, and then the other letter is unknown, and we're trying to solve for that unknown letter. But we know that because it told us that H is dominant, then obviously we'll put the capital H as stated.
Yeah, because we know that that individual has one big edge at least, because they have the dominant phenotype,
Yes.
right?
Yes.
Okay, good. Okay, so can we go back to this question now about the this one? You all followed this one, how you use the product rule to get the
Yes.
offspring of this expected genotype. So this is a different one. This was the one we were doing, right? So 1 out of 64. Okay, so that's for the genotype. But the next example is for the phenotype. And I'm not going to erase that, just we will be able to use that same information, just use a different color, so I'll be able to hopefully separate them out for you. Now, we need the same cross, it's the same cross, so we use the same information of the 4 individual crosses, but how many progeny will show the dominant traits of A, C, D, and recessive for B? So they're asking us A, B, C, and D. They're asking us this. Sorry, you can't see that, right? They're asking us this. Whatever is in blue. Look at whatever is in blue. Okay, I'm going to erase this one. Okay, I'm going to write it up again. So they're asking us dominant for A, recessive for B, dominant for C, and dominant for D. This is what they're asking us. So then, you know, dominant for A is this, whatever I've put it in blue. Recessive for B is this. Dominant for C is this. And dominant for D is this. Right? You can see all 4. So that's going to be 3 out of 4 cross to 1 out of 4 multiplied by 3 out of 4 multiplied by 3 out of 4. So this is going to be 3 times 3 times 3 is 27 divided by 4 times 4 times 4 times 4 is 256. Okay, got that? Because we want big A, little a. When big A, anything, so that's why we use this one. Little b, so that's why we use this one. Big C, anything, right? So that's why we use this one. And then big D, anything, that's why we use this one. Okay, everybody with me? Okay, one more poll question. I'm hoping that these poll questions are helping you to understand and at least gauge whether you are following or not. So I'm going to launch this poll. I'm going to give you some time to do it. Again, the uppercase letters represent dominant alleles, the lowercase letters represent recessive alleles, and so try to do this. In peas, seeds can be round or wrinkled, yellow or green. Stem may result from Stem length may result in a tall or dwarf, big T or little t. And so we have three traits here, and this was your cross. What proportion of offspring would be tall plants with round yellow seeds? Now erase this, we'll see what that comes up. Try to work on it on your own, and then I'm going to show it to you. Okay, still a few students have not done it. I'm going to give you until 4 minutes, okay? Just 15 seconds, and I'm going to stop this. Okay, I'm going to stop it. I'm going to share the results. A lot of you got it correct, but a lot of you did not. The correct answer is A, 1 out of 4. Okay, and let's see why. So I have it written down here. So you have this cross. Big T is dominant, big Y is dominant, and big R is dominant. So you have that cross, which I wrote up there, and I separated each individual cross. Big T, big T, big T, little t, cross to little t, big Y, cross to big Y, and big R, cross to little r. I wrote those three down, and then I wrote down what we need. We need what proportion would be expected to be tall, round, and yellow. So it's big T, and it can be anything, big R and anything, and big Y and anything. So you know that for each individual crosses that we did, big T, little t, cross to little t, you will get 1:1 ratio of big T, little t, little t, little t. So 1:1 of tall and short. All are going to be big Y because that's what the parents are, homozygous for the big Y, big Y. And then big R, little r cross to little r, 1:1 ratio of big R, little r, and little r, right? So therefore, to get this, you have to have 1 out of 2 of the big T, 1 out of 2 for the big R, and 1 out of 1 for the big Y. So it's 1 out of 4 total. Everybody understood this? Questions? No, if I relaunch the poll, then the old stuff goes away. So it's okay. Don't worry about it. If you miss a poll or you get it wrong, it's okay. A few polls that you missed throughout the semester is not going to win your participation points. Okay. All right. Everybody followed this, or you have questions about it? Okay, seems like everybody followed it, right? Okay. So just a little bit of historical information. You will get credit if you answer it wrong. Yeah. You will get credit if you answer it wrong or right. You will get credit as long as you participate in the poll. Okay. And what I'm going to do is I'm going to give you so many polls throughout the semester, and at the end of the semester, I have a way from Zoom to retrieve how many polls you participated in, and I'll just give you points according to what your participation is. Okay? I'm not going to look at your answers, so don't worry about that. But I want you to think about it and answer, because that gives me some idea about whether you're following or not. Okay. So this is just a little historic slide that tells you that when Mendel did his work and he published it in 1866, very few people actually believed him. And he did so much work and so much methodical research, but always research has to be repeated, reproducible. And so scientists did his work again to independently check whether his crosses and his ratios were what they also got. And it took until 1900 for 3 scientists to independently work on their own on these crosses and validate Mendel's work. And therefore, eventually, he got credit for all the work that he had done. So now we'll talk about genotypic classes, which is a slightly different concept, because we've talked about it all along. We've just not used these terms, genotypic classes. Okay. So when you do this cross, I'm going to do AABB for simplicity. AABB cross to AABB. Okay, you do this cross. You know that you will get a 9:3:3:1 ratio of A. These are your 4 genotypic classes. Okay. The 4 phenotypes. These are, the phenotype is A and B. The phenotype is A and B. The phenotype is A and B. Whereas uppercase letter represents dominant phenotype, lowercase letter represents recessive phenotype. So these are your 4 genotypic classes representing the 4 phenotypes. Okay, everybody with me? So the way you can actually calculate this for a dihybrid only, having to not go through the Punnett square, is we'll have to break this up. So this one here, if you look at the 9 ones, so AA is equal to AA how do you figure out how many of each do you get? Okay. These are the four types. These are the four types. From A dash B dash, you can get these four types of genotypes. Okay, how many of each of these would you get? To calculate that, there's a simple method. So here you have a homozygous and a homozygous. So it's 1 times 1, which is 1. Here you have a heterozygous, so it's 2. And here you have a heterozygous, so it's 2. So it's 2 times 2, that's 4. Here you have a homozygous and a heterozygous, so it's 1 times 2, that's 2. A heterozygous and a homozygous, so it's 2 times 1, that's 2. That makes 9. Okay, so that's how you calculate how many of each of those four genotypes will you get for that genotypic class. Genotypic class being dominant for A and dominant for B. Okay, is everybody with me? So then let's do the second one, which is this one. A, I'm going to pull it up. A, B, B. There are two possible outcomes, A, B, B, or A, B, B, right? Both of them are homozygous. So here is homozygous. So it's 1 times 1, it's 1. And this is heterozygous. So it's 2 times 1 is 2. So that makes it 3. So that's here. With 9, 3, 3, and 1. We do the same thing for the other two as well. Okay. Are you following? So this is just applying to the laws of probability to figure out whether you have how many of each genotype do you have for each genotypic class. Okay. Okay, going forward, we just have a couple of minutes. Multi-hybrid crosses results could be predicted based on the rules of probability. We saw that already. And separate individual traits and calculate probability for each phenotype and genotype. We saw that already. We did that already. Hopefully you know how to do that. And you can combine the traits. Okay. Two general molecular principles. A specific gene determines a specific protein whose activity may affect the phenotype. So what is the rationale for dominant and recessive? So if you have a specific allele, you have a specific type of protein being made that results in a specific function. So if you have a dominant one, let's say you have a dominant for eye color, it makes the pigment, and so you have a dark colored, black colored eyes. You have the recessive one, it makes less pigment, you have the light colored eye, right? So that's how the protein does the function that results in a phenotype. Usually the dominant is the wild type or the normal, and the recessive is usually a non-functional or lack of function or less functional outcome. Normally, but it doesn't have to be that way for everything. There can be exceptions. Okay, time's up. I'm going to start with this fall in the next class, and then we move on to the Mendelian inheritance in humans. Okay, we have a couple of calls and then we go to that one. I'm going to stop share, and then let's see if you guys have any questions. I can answer them quickly before we stop today's session. Okay. Questions, anybody? How did today's lecture go? Can you all hear me clearly?
It was much better, Professor.
Okay, good. So these settings work. Nice. Anything else?
Professor,
Yeah,
I have a question.
sure.
So for the week, not the weekly quiz, but for the PLF quiz, is it due on Friday, or is it during the PLF session that we are attending?
Yeah, for the PLFs, you have to ask Armin. Armin, are you here?
Yes, I'm here.
Armin, can you answer his question, please?
Hi,
Yeah,
Armin.
hey there. It depends. Sometimes the PLFs go for a long time, so those times I allow you to just take it home. But usually we leave some time towards the end, a good 20-30 minutes, so you can take them. Sometimes depends on the time.
Oh, sounds good.
Are
I
the
just saw that it was due on Friday. That's why I was asking, was it due at the end of PLF or anytime?
Yeah, this week is gonna be due Friday.
Okay. Thank you, Armin.
Are the PLF quizzes graded for accuracy?
Yes, you're graded, and whatever it's on there, we usually cover it during the PLF session. So as long as you're paying attention, you'll do fine on the quiz too.
Thank you.
Okay, so for the recordings, I've set up so that you can see on the right-hand top the screen, either the whiteboard or my face. So hopefully, let me know if you can see that whiteboard, okay, in today's recording. I've set it up so you should be able to see the video. All right.
I have a question, Professor. You said that to remind you about activating to show what answers we got wrong on Connect.
Yeah, I'm going to try and go back to Connect and see that.
Okay.
It probably have to be tomorrow morning.
Okay. Thank you.
All right.
Have a nice day.
You too. All right. If there's no other questions, then I'm going to end the meeting. If you have any questions, stick around and ask me, okay? I'm going to stop.